Good morning, everyone. We might get started with today's science seminar, which is hosted in Jewel by the Accelerating Crop Genetic Gain IRTG and also the ARC Training Centre in Predictive Breeding. Start off with an acknowledgement of country. The IRTG for Accelerating Crop Genetic Gain and the ARC Training Centre in Predictive Breeding acknowledge the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognise their valuable contributions to Australian and global society. My name is Richard Dixon. I'm a PhD student here at the Centre of Crop Science at QWOFI, and I've done quite a bit of work with our speaker today, running GM field trials just outside of Adelaide. That's why I have the pleasure to introduce this morning. Today is just a bit of housekeeping. Today's seminar is scheduled from 11 a.m. till 12 p.m. We'll have around a 45-minute seminar followed by a Q&A session. During that webinar, if you have any questions, please submit them via the Q&A tab rather than the chat function so that we'll be able to see them and address them to Stu. And just critically, anyone in the room who's asking a question, if you could just wait till the microphone comes to you so those online are able to hear. So our speaker today is Professor Stuart Roy from the School of Agriculture, Food and Wine at the University of Adelaide. Stu's a professor at the school. He's currently the Deputy Director Industry for the ARC Industrial Translation Training Centre for Accelerated Future Crop Development on the Management Committee for Plant SynBio Australia and formerly the Director of the ARC Industrial Transformation Research Hub for wheat in a hot and dry environment. His interests are in improving the abiotic stress tolerance of cereal crops, particularly salinity tolerance, and improving cereal yield. So I'll hand it over to you, Stu.
Great. Thank you very much, Richard. It's great to actually get up here to be able to sort of talk to everyone about sort of the research that you're doing here, and also the research that we're doing sort of through the training centre. What I've got today is a bit of a Frankenstein presentation here. I want to go through an older project that we've had running for about 10 years in the centre, and this is formally an International Wheat Yield Partnership project. But at the same time, hopefully, as you see sort of in the audience from the fantastic work you do here in both your training centres, how it could be done a lot better. Now, there's a lot more that we know about breeding, around sort of the use of gene editing and sort of getting things out into the field as well. So as you go listen to the presentation, hopefully you can sort of see the progress that we made, but also now how things can be done much better with all the knowledge and things we've got today. I think the original plan for today was, I said, 3 genes for high yield. In the interest of time, to keep it down to 45 minutes, I've removed a gene. So we're just going to be looking at 2 genes that have come through from the IWIP program. So just to give you a little introduction to the International Wheat Yield Partnership, this was a conglomerate of research groups from around the world, overseen by the IWIP committee. And the goal for that organisation was to look at increasing wheat yield by 50% over a 20-year period. The drive for that is that currently with wheat worldwide, the annual increase in yield achieved through current breeding programs is less than 1% a year. In Australia, we actually do fairly well in wheat. Our breeders are able to improve it by about 1.2, by 1.6, but it's still a lot lower than trying to this 50% yield in target. So what IWIP was designed to do was to fund a range of high-risk projects, acknowledging that most of them wouldn't necessarily deliver something to breeders, but that 1 or 2 of them would actually pass something on to the Maize and Wheat Breeding Institute at CIMMYT in Mexico, and also to breeding programs within the different countries. So our program, very much our project in this, was very much one of those high-risk projects. It was involving doing GM wheat, which at the time the project started in 2016, there wasn't any GM wheat commercially available around the world. Now we've got HB4 wheat, which is a GM wheat released in Argentina and Brazil, and that's currently undergoing controlled field trials in Australia. But when this project started, it was GM, it was high risk from that point of view. It was also high risk from the genes that we were using in the project. So to introduce you to the 2 genes I'm going to talk to you today about, we've got the Arabidopsis vacuolar proton pumping pyrophosphatase gene, or AVP1, and the rice phosphorus starvation tolerance gene, PISTO1. So What these genes do is they encode proteins that are very helpful to plants for their abiotic stress tolerance. So the AVP1 gene was originally identified in Arabidopsis, and is a protein that's found on the tonoplast of the vacuolar membrane. And what it does here is it uses the high energy molecule PPI, pyrophosphate. It breaks that down to pump protons into the vacuole. And then through that secondary process, what that does is that puts a lot of protons in the vacuole that other transporters, other symporters or antiporters can use to move ions across the tonoplast membrane to keep them in the vacuole. And what it was originally discovered for was because it had very good properties for enhancing the salt and drought tolerance of any plant that you overexpressed this gene in. What would happen is you do overexpress this gene in, say, Arabidopsis here, you'd end up getting more protons into the vacuole, then the plant would be able to transport more sodium into the vacuole and make the plant more salt tolerant. Similarly, by building up a high proton gradient in the vacuole, you're able to generate a lot more turgor in the plant cells, which allow more water uptake coming in. And plants overexpressing this gene would have a greater drought tolerance. And we played with this in what was formerly the Australian Centre for Plant Functional Genomics in the salt group there by expressing this gene in barley. And when we express this gene in barley, we're able to enhance the biomass of barley plants in the greenhouse. And when we translated that out into the field, into very high saline field sites, we can actually see the salt on the surface here in the trial. These are these are expressing lines of the AVP, and this is the wild type plants here. And as you can imagine from something that looks like this, only the transgenic lines actually produced any yield. Before I go further, I just want to sort of point out a couple of things just more generally from this. When we're working with transgenic material, we're always working with a number of independent transformation events. So when you see graphs like this, you'll see that these are 4 different independent transformation events. And the reason we do that is because when you insert a bit of DNA into an organism, the position that DNA goes into varies from event to event. And so if we want to try and show that something is GM, actually has a beneficial phenotype, you don't just want to do it once, you want to do it multiple times to sort of see the same event in there. The other thing to point out that you'll see in all the graphs that we've got as well are these null segregants. And this is what we compare the performance of our transgenic lines to. These were formerly transgenic plants that through the process of going through different generations, they've actually lost the DNA in there that made them GM. So they've effectively reverted back to wild type, but they have the same history as the transformation as the transgenics. So they've been through the same transformation procedure, they're the same cultivar, they've been through the same greenhouses, etc. So we're comparing effectively like with like, the only difference being these guys have our gene of interest and these guys have lost it. The other gene that we were working with is this rice phosphorus starvation tolerance gene, PISTO1. This is a protein kinase. It was identified by Sigrid Hoya when she was working at Erie, and then she brought that research to the Australian Centre for Plant Functional Genomics for looking to see how that could improve the phosphorus uptake of wheat. Now, it was originally identified from sort of mapping populations and GWAS populations where rice that was rain-fed and from sort of drought-tolerant lines had the ability to have a greater uptake of phosphate from the soil. And QTL mapping for that identified a region which was called PUP1, where the plants that actually had the beneficial trait produced a lot more roots and had a greater sort of root biomass. And the hypothesis was that these were then better at uptaking phosphate from the soil. Sigrid and her group then generated transgenic rice that were overexpressing the PISTO gene, which was positionally cloned from the QTL. And this is just an example of one of the transgenic rice lines along with a null segregant growing under low phosphate conditions. So you can see the transgenic did really well, whereas the null segregant didn't. And again, these plants had much greater root biomass than the null segregant. And in the rice, no matter whether you grew the plants under low P, which is in red here, or under high P, which is sort of in the purple there, the transgenics had a greater yield in the greenhouse. So why were these good for IWIP? So talking about enhancing the stress tolerance of either the drought tolerance or salt tolerance of that barley or the phosphorus use efficiency in rice. IWIP's looking for optimal conditions. But what we actually found when we put this material into wheat, or when we're working with our material in other species, is that the transgenics not only had a beneficial phenotype under the stress that we're exposing them to, but they also had a good phenotype when the stress wasn't present. I'm just going to show you this for the vacuolar pyrophosphatases, but it was the same for the pistil as well. So here's the barley that I showed you earlier, that was showing a greater salt tolerance and yield in the field. Here's that graph from the greenhouse that's been redrawn to include the low saline, no saline plants that are growing in there. So you've already seen the grey bars. That's the performance of the transgenic lines under salt stress. There are null segregants here, and then an improvement in biomass production in these plants when salt's present. But also under optimal conditions where salt wasn't present, so we didn't add any additional salt, these plants had a greater biomass in those conditions as well, which when you put them out into the field and ran them in the field, there was a greater yield of seeds from each plant that was in there. So the vacuolar pyrophosphatase is actually helping the plants grow better, just generally, whether salt was present or not. And it was the same with the pistils. And it didn't seem to matter whether we used the Arabidopsis version of the gene or if we used the barley version of the gene. We always got this sort of enhancement in biomass. So what we want with the IWIP program was, well, what happens if we translate this into wheat? Can we take the vacuolar pyrophosphatase gene? Can we take the protein kinase, transform them into wheat? Do we see an improvement in the performance of these plants? Why are the plants doing better? Can we get a handle on some of the mechanisms behind it? And then, because at the time there was no obvious pathway to market for a GM wheat, can we come up with a non-GM equivalent that we could effectively pass on to the CIMMYT breeding program and to the Australian breeders? So we transformed the Arabidopsis AVP gene into wheat, and we saw very promising results of that under greenhouse conditions. We saw a greater shoot and root biomass in the plants. So this is the root data here, where the transgenic lines have a greater root biomass, and that's translated into improved, I'm going to say, yield in the greenhouse, because a breeder normally slaps me around the head whenever I talk about yield in the greenhouse. But they did result in these lines producing more yield. We sent this material to our collaborators in the IWIP program at the University of Arizona, Roberto Vacuolar, and they started to look into the mechanisms behind what was going on here. And what seems to be happening is that in addition to the ability for these plants to actually be able to uptake ions and secrete them in the vacuole, which helps generate turgor, the transgenic lines also had a greater ability to move carbon from source to sink tissues. So moving from the leaves into either the roots or into the grain. And Roberto and his lab found that this was because the vacuolar tonoplast protein actually has a different role depending on what cells it's in. When it's in a leaf cell and in sort of the mesophyll, it's on the tonoplast. But what he was able to show is that in the cells around the bundle sheath, where you're looking at moving sugars into the phloem, that the tonoplast vacuolar pyrophosphatase was actually in the plasma membrane. And then Roberto was actually able to get in and do some targeted knockouts and actually show if you disrupted that process, you stop the movement of sugar going into the phloem, into the grain, into the leaves. And it looks like, in this case here, not just being expressed on the tonoplast, but the vacuolar pyrophosphatase is actually on the plasma membrane, potentially working backwards. So rather than breaking down pyrophosphate to pump protons, it's actually synthesizing pyrophosphate, which gets used in other processes, which then enhance conditions for sucrose transport into the phloem. So that was why these plants seem to be growing better and sort of having improved biomass and yield. With the pistil, we also got some very promising results from the greenhouse, and I apologize how the graphs are different between the pistil and the AVP, just different we presented these in the different publications. But what we got from the pistil, as you can sort of see from the greenhouse pictures here, is that the plants expressing the pistil-1 kinase had an improved improved biomass improved number of tillers and heads being produced on them, and also had a greater time to flowering. So what this diagram sort of here is showing is the blue lines in this are null segregants, and the green and the red lines are transgenic material. And you can see that we've got the transgenics with a greater amount of biomass, a faster time to flowering. They were 10 days faster to flowering, but actually produced more seed per plant, more grain per plant under the greenhouse conditions. We collaborated with Julia Berry series across at UC Riverside, and the master's students in Julia's lab found that these plants, a bit like the rice, had a lot more root biomass than the null segregants. So here are nulls across here, and here are the transgenics. And that in these transgenics, there was an upregulation of the phosphate uptake pathways in these plants. That's what you're sort of seeing in this sort of plot here. So these plants were able to upregulate the processes that were bringing phosphate from the soil into the root and through the plant. We also found, and that's sort of shown in these diagrams here, this is looking at proton efflux from the root, so the ability of the plants to acidify the rhizosphere. So the blue lines are our null segregants, and our red and green lines going down this way is sort of the protons coming out of the roots and acidifying the rhizosphere. So the transgenics were able to acidify the rhizosphere to help with the phosphate uptake. So in the greenhouse, it looked like the AVPs and the pistil in those environments were enhancing biomass production and also yield. But just because it does well in the greenhouse doesn't mean it does well in the field. And anytime you're doing translational research, you need to be able to sort of show that you do actually see those beneficial phenotypes in the field. And although we were with a GM project here, we still wanted to show that manipulating the expression of these genes did actually lead to an improvement of yield in the field. So we wanted to get out into doing field trials. So the reason for doing field trials, obviously, the greenhouse environment is very, very controlled. You are basically looking for the effect of your phenotype on the performance of the plant, and you're trying to maintain everything else to reduce confounding factors. But when you're in the field, that's when you really get the opportunity to see how your material performs under different seasons, under different environmental conditions, under disease pressures, you name it, out in the paddock. And so anything that we sort of take out there, we want to sort of be able to see, does it actually perform like it does in the greenhouse? And could that then be useful for including in a breeding program? So with our GM stuff, or if we stick out gene edited material in the field, we wanted to confirm the performance of the phenotype, performance of the trait in the field. We also want to be looking for the agronomic performance of the lines to see whether our material is actually forming agronomically similar to the current elite cultivars that are growing out there. Potentially, if we do get down to that stage, even looking at things like grain quality and things like that. We're also looking for identifying off types that you might not actually see in the greenhouse in terms of the plants the seeds shattering in the heads, or having plants that are a little bit taller than they're meant to be, that sort of thing. Anything that could be detrimental to sort of breeding. Now, characterization of biotech plants in the fields, challenging. Having a chat with Ian about this earlier on today, and the fun and games that you have, and actually getting approval for doing the regulation, and then also maintaining the sites in a way that you don't accidentally have unintentional release of your material. So in Australia, GM field sites are regulated by the Office for Gene Technology Regulator, and you have to apply to them for very, very specific licenses for growing plants out in the field. And the regulator is very concerned about accidental release of material into the environment, and also implications on human and animal health. So the licenses that you apply for the regulator are very specific. They're very specific down to what gene, what cultivar, what promoter, terminator you actually put out into the paddock, and they will evaluate your application and then give you a range of license conditions which minimize the chance of the spread of your GM events out into the environment. The process takes about 9 months to get the license from the regulator at this moment in time, although they are reviewing the rules, and that might dramatically sort of decrease. And the license conditions do vary from license to license, but are very specific in what you can do. And quite often, what you have to do for growing the field trials isn't necessarily the most optimal way for doing field trials in terms of time management, because the OGTR are not really interested in you performing optimal trials. They're more interested in stopping material getting accidentally released. So they are very expensive to run in comparison to conventional trials. So we've done some calculations in the past just to give you sort of an idea, and I can get the Adelaide uni breeders to give me non-GM trials where it costs me about $25 a plot to do the trial. Comparing that to the stuff when we go out and do field trials, because of all the regulatory stuff on top of that, we're anywhere between $300 and $1,000 a plot because of the conditions. A lot of these conditions are down to how we actually maintain the field site. So we've got to, in addition to actually growing the plants out there and looking after the plants, we've got to make sure that the different zones of the field site are maintained in such a way that we can spot any escapes of genetically modified material, but also any sexually compatible plant species that are growing near the site where you could get accidental gene flow from. So we've got to do things like have a monitoring zone 10 meters around our trial area that's effectively kept scorched earth, so nothing actually growing in there. So a lot of our time's actually out there weeding the trials. We've got to maintain an area around the outside of the GM field site where the grass is going to be kept, at least for wheat and barley, below 10 centimetres, and we've got to constantly monitor that for any sexually compatible species popping up in that area. We've got to make sure we've got containment and tracking processes. Often it's actually easier to do things by hand than it is to use machinery because of the clean down processes you've got of the machinery. And I want to show you this because this was our original GM field site in South Australia. And the area of the trial itself, which was in sort of here that we could grow GM plants, was only a quarter of a hectare. Our original trials we had out there were only 4 meters wide and 17 meters long, but the area we had to monitor and inspect frequently to make sure nothing was there was about 12 hectares. And so you can imagine going out to that paddock, we'd get about 8 to 10 people in a police line. I don't know what it looked like to the general public, could wander past when we're doing this, and basically walk through that paddock, making sure we covered that area, that nothing was sexually compatible within that when the plants were growing. Not just when the plants are growing, that when you've done a field trial, such as this one here, you then have to monitor and maintain where you've planted for a minimum of 2 years afterwards. At the same time, also maintaining the field site area. So a 1 year trial actually converts into 3 years of monitoring of the trial site and managing the trial site. So in addition to everything costing, a lot more expensive, it's a lot of people hours actually doing it. And we worked out for the 2018 results I'm going to show you here, it was more than 6,000 people hours actually out in the paddock in terms of doing all the compliance monitoring in addition to the actual plant phenotyping. And that for that 1 year trial was effectively for the 3 years involved. So with the costs of doing GM field trials like this and all the regulatory things, we've tried to look at a way of making this a bit more cost effective and also opening it up so that other research groups can come in, and if they've got any material they want to put out in the paddock, to actually have it in such a way that they can come out and use it without having to have quite literally hundreds of thousands of dollars to actually manage and run a field site. So we've, over the last few years, developed a field site out at the bottom of the Barossa Valley called Rosedale. It came out of necessity because the South Australian government reacquired the land that our GM field site was on, or small GM field site was on, and we had to come up with a new field site. And we thought, well, while we're doing this, let's actually try and future-proof this and have it as a facility where other people could come out. We have dedicated farm equipment out at the site. And also new ways of phenotyping the site. So we were fortunate enough to get investment from a range of sort of universities, a LEAF grant from the Australian Research Council, and also a GRDC infrastructure grant to build a 2 hectare area underneath bird netting out at Rosedale. We've been able to purchase farm equipment, harvesters, tractors, you name it. It's not sexy science equipment, but it's equipment that stays on site. that can get cleaned on site, but then never leaves the site. So you don't accidentally have seed caught up in your harvester, leaving your site and then ending up in somewhere where it shouldn't be. We've been able to install in the site, thanks to working with the Australian Plant Phenomics Network, a range of range of cameras, fixed and mobile, that can point at the plots for people to actually see how their plants are growing, but also for compliance and monitoring. And we were able to buy in a prototype robot, sort of seeing here, which we've kitted out with a hyperspec, a LiDAR, and an RGB camera that you can actually have the field site and run it across the trials in the paddock. The idea being giving people that do the stuff in the field site the ability to actually use some more high-tech equipment rather than just sort of a measuring tape as they go through. And working with the APPN, they're in the process of helping us kit all that out. And they're actually going to develop 12 of their own versions of that robot for sending out to the APPN nodes for people to use on non-GM trial sites. So this is a new facility. We've worked with this with Richard and Lee last year. We've had some of their material out. We've done 3 years worth of trials, and we're now at the stage of actually passing this facility on to Plant SynBio Australia. So they'll actually take it off my hands, so it's not my lab running it, and they'll actually do all the monitoring with the hope that next year I can come along and say to them, I just want a bit of space, in the same way I go to the APPN and say I want a greenhouse, and they'll help actually run and manage it all for me, and I can just focus solely on working with the plants that are in there. One of the other things we've been doing through the training centre is actually trying to help make the compliance activities that you do at these field sites easier to do, and starting to use AI to help with some of that processes. So one of the common license conditions you get from the regulator is that you have to let the regulator know 14 days before your plants start flowering, and you have to start doing inspections of your monitoring and isolation zones. So depending on the crop, going up to almost 1 kilometre outside of the field trial area, looking for sexually compatible species. Actually going out and checking the development of your plants and seeing how close they are to flowering is fairly time consuming in itself. And what we had at the training centre is Yatin Z, who's just our first student from the training centre, graduated, is that Yiting actually developed a program, some software for actually looking at the plants as they were growing in the paddock, and working out the developmental stages, not for a phenotyping purpose, but for a regulatory type of purpose. And so what you've got here is Yiting, this is him trying it out at a non-GM field site, deliberately, because there's lots of weeds in the background, so we wanted a lot of noise in the background. This is just footage taken with an iPhone and then sent back to his algorithms. And the computer is able to then identify various stages of the plant development, and then combining the different developmental stages with data from previous years of how things performed, to then also looking at the long-term weather forecast for the area, is that Yiting was able to come up with a software package that would effectively tell us when our plants were 21 days away from flowering, or a maximum of 21 days away from flowering, between 21 and 14 days, which will allow us to get out to the paddock and actually start doing those flowering time inspections and allow us to meet our OGTR license requirements. And he's now done this for wheat and barley. He's also got it going for canola as well. And things like that are going to help us actually monitor the sites a lot better. So that's GM field sites, and we were able to get our IWIP material out into the paddock. Now we've got effectively 3 years worth of data from 4 sites. The 2018 is the site that was our former site south of Australia. That was the very small plots we used in there. 2023, 2024 were at our new facility, and 23 we were able to get across and work with Alex Johnson at the University of Melbourne and get into his field site at the Dookie campus. And what we were able to see from this material was we were able to get a translation of what we saw in the greenhouse to happening in the paddock. That we were getting yield improvements from the transgenic lines in the field, and depending on the type of year we got, the Pisto or the AVP, one of them would do better than the other. If it was a very dry year, such as in 2024, when we had about 25% of the rainfall that we normally get from January to June. And then this was the rainfall in that season. So the green line is the long-term average. And that's the rainfall we got in that season. Basically, it was about 30% of the long-term average we got. The AVP plants were actually still able to perform and actually have a yield improvement in those lines under those conditions. The pistil plants did very well when there was lots of water around. In the 2018, the 2023 seasons, they actually performed really sort of well under these conditions, but did not do so well when water was limited. So the nice thing from this is that the transgenics were actually showing a yield improvement in the field. So the candidate genes were possible things to chase for the IWIP project. And depending on whether you're in a well-watered region or in a drought region, you would pick pistil or you'd pick the AVP. So the transgenics are showing something promising. Can we go about this in a non-GM way? Because at the time when this project was originally conceived, there was no HB4 wheat, there was no commercial GM wheat out in the environment, and it was looking like Australia wouldn't have any commercial GM wheat. Now there is GM wheat undergoing limited field trials, and possibly that might happen in the future in Australia. So the approach that we took to go about it in a non-GM way was effectively the transgenic lines were overexpressing or had a high expression of a transgene. Could we then identify the wheat equivalents of these genes? Could we then look for wheat equivalents that already had a high expression and just breed them into our current cultivars? Or could we take more gene editing approach and look at enhancing the expression that way? So first of all, we had to find the wheat equivalent of the genes. So we looked at the different pan-genomes that were out there and identified that bread wheat had 15 of the vacuolar pyrophosphatase genes, and that had 6 of the pistil, the protein kinase genes in there as well. We then did a haplotype analysis to work out how many different types of versions of these genes that we had. And then we also looked at the predicted protein sequences and the expression profiles of the wheat genes to find the most related pyrophosphatase gene to the AVP and the most likely gene that was most similar to the rice pistil one as well. And so just for showing you this for the AVP, we were able to find, depending on the gene, between 3 and 14 distinct haplotype groups for that gene. We then did some expression profiling across a range of diverse wheats for the different haplotypes, and we're able to find that TAVP2D in bread wheat was the one that was most similar to the AVP in terms of the timing of when it was expressed and the tissues of where it was expressed, and the sequence of the protein as well. And then when we actually looked across the different haplotypes, or sorry, the different cultivars out there, we've got some of our standard lines, we've got Mace here, we've got Young and Scout, and then we've got some of these accessions here which have 3 to 4 times the expression of the AVP gene. And we did the exact same thing with the pistil. So the next few years after that, we're done actually trying to integrate these into Scepter, one of Australian Grain Technologies lines, to actually see if we could get an enhancement of yield that way. And to skip over quite a few years of testing in the greenhouse and a few sort of results in the greenhouse, and this is what we got from last year's trials. So very much preliminary data, 1 field site, 1 year, and don't read too much into it. But we have been able to introduce in the top graph here a novel allele of the pistil gene from an accession called ROLF207. So this is Scepter's performance here, and then this is independent lines where we've introduced that allele into those lines, and we see sort of an improvement in that one. We had a little bit of a stuff up of bulking in 2024, so we couldn't put everything out into proper large breeders' plots like what this was done, and so we had to stick them out into smaller, some of the material into smaller plots. And this material here has got some of the pistil alleles, but also some of the vacuolar pyrophosphatase alleles that we suggest we were looking for there. And again, some sort of nice preliminary data for that material. And where this is going now is the project we've got for this has two industry partners in this. It's got AGT, it's also got Longreach. They are going to be planting that material around around Australia this year in 12 sites in total, 6 AGT, 6 Longreach, and that's where we'll actually start getting a little bit more information from these lines as to whether or not this is actually reliable, repeatable, and actually doing any good. The other way, of course, of going about this is gene editing, as you're probably aware, certain forms of gene editing are considered non-GM in Australia, and so that approach is called SDN1, which I'll touch on in just a second. And for that, what we're looking to do is looking to get into the promoters of these genes and actually knock out negative regulators of expression. And in both the pistil and the AVP genes, we've identified what those negative repressors are in the promoter regions, and we're in the process of actually generating some of these plants now. So hopefully we can go about this in a non-GM way. That kind of leads into the last part of the talk and just introduce you to one of our other PhD students from the training centre. And that's looking around gene editing and whether it's going to be considered GMO or not GMO, and the grey murky area you get in terms of actually being able to translate some of gene edited stuff into stuff that people will grow in the paddocks. So I just want to give a very quick refresher on gene editing and where the OGTR currently sits on gene editing in Australia. So we have a number of ways of going about doing gene editing, whether it's talons or zinc finger nucleases or CRISPR. They all tend to work in the same way. You have a nuclease which you target to a very specific region of your DNA, which cuts open the DNA. And then you can either let the DNA repair itself, which can introduce mutations into the DNA, and that's what's typically called SDN1. Or you can guide the repair of the DNA either by guiding nucleotides in there, what's going to be in there, or inserting in whole genes into the genome. And that's typically called SDN2 or SDN3, depending on the size that you insert in there. Now, the current OGTR regulations for gene editing say that SDN1 is considered non-GM. And SDN2 and 3 are going to always be GM. Now, these definitions are all different in different countries. That's something else you've got to be aware of as well. But in Australia, SDN1 is non-GM. However, the most easiest way of producing a gene-edited plant is to actually go through a GM phase. So to get the enzymes and the guide RNAs that are responsible for making your edits in there, the most common way of doing this is to insert a DNA construct into your plant that's got the gene for the protein, for the nuclease, and also your guide RNA. And so that individual plant is GM. If you've got a plant with an SDN1 edit and that bit of DNA still in there, it is still GM, not because of the edit, which the OGTR say is not GM, but because this still sits in there. Now, the regulator has said if you then do crossings, and you can sort of cross with plants or do some selfing, and you end up segregating the lump of DNA that's got the Cas9 in it from your edit, this plant here will be non-GM, and this plant will still be GM. And this now is not part of the OGTR's interest, and you could go out and stick that into non-GM trials. However, the regulator has said that if you're left with a tiny bit of DNA in there that shouldn't be in there at all, then it would be considered GM. And where the breeding industry and our university in particular are very, very paranoid at this moment in time is, what if this scenario happens? Where, yes, this is definitely a GM organism, and it's got the SDN1 in there, and we can detect that fairly easily by doing PCR, southern blots, or just even growing the plants on selectable markers. But what if we end up in a situation where we have a little bit left of this fragment in there? According to the regulator, that's GM, but we might not be able to detect that. Europe said anything less than 20 bases is going to be fine, it's going to be non-GM. Again, how do we go about detecting this issue here? And is it actually a problem? Well, we've already had this happen before, and I've been chatting to some people earlier around this as well. And you're probably aware of the polled dairy cattle that were produced in the US, where they used a talon approach to edit the cattle to remove the genes where the cattle produce horns. And that was all great. And a breeding program set up in Victoria in Australia, where they brought in semen from the cattle there and used a sort of a breeding program here. And the cattle that came from this was, with hornless. The problem was another group at the USDA, completely independent, came and sequenced the US cattle and actually found there was a small fragment of bacteria DNA left in the genome, which then made these, which were considered non-GM in the US, GM, and completely scuppered the breeding program that you had in Australia. And that's a concern that the breeders currently have with this setup, is that as sequencing technology and things get better over time, if a mistake is made now and the cultivar is released and then it comes back later on and finds it is GM, they're worried about the replications coming back to them later. So we've got a PhD student, Kira Danes, at the training centre, who's trying to come up with a way of being able to screen gene-edited material to show that it's free of any GMG contamination. Large breeding companies can afford to do whole genome sequencing, platinum-style sequencing, looking for these introduced fragments. It's less practical for a research lab to do this. And so Kira's looking at a way of actually trying to identify any fragments that are left in the genome. Now, the first thing that she did was she went to see, well, let's actually see how bad the EU description of a GM event is. The EU said anything 20 bases would be considered GM. So what Kira did is she took 3 of the most common vector backbones that you have that we transform wheat and barley with, the PMBC, the PANIC, and the P-direct. And she split them up into 20 base chamers all the way across, and then went into the sequenced Arabidopsis, wheat, barley, canola, rice DNA for non-GM plants, and to look for how much of these DNA sequences are already in the plant, i.e., could you accidentally identify a non-GM plant as a transgenic plant? And of course, the answer is yes. There are multiple parts of our plasmids that get put in there that already exist in the DNA of our non-GM plant, including regions, 150 bases in size from the vectors that are actually in there naturally. It's not GM, it's just naturally being introduced into there. And so what Kira's now trying to do is come up with a better way of, actually identifying what does make a plant GM and what doesn't. And she's also talking to the regulators here in Australia just to try and get some better guidance on how that kind of goes. What are the implications? Well, this is a worst case scenario that we use in our training centre when we talk about stewardship and what happens if accidentally I've just provided non-GM seed to AGT. What if I accidentally contaminated that seed and provided AGT with GM seed? These are the possible implications that you would have of that happening, and could involve, in the really worst case scenario, completely stopping your university from being able to do any GM stuff. So that would also be other schools, having to give money back to the funders, having to pay for loss of revenue for farmers, your VC, DVCR going to jail, and this is what's keeping our regulators at the university up at night. And it has happened. You've probably all heard about the sorghum story, where sorghum was brought in from the US that was accidentally contaminated with GM seed, and that actually made it into seed, the Dominator line that was released out in the public. And PB Agri-Foods have had to basically call back all the Dominator seeds, all the Dominator plants that were growing in there. We had some Dominator down at the University of Adelaide. It was part of a breeding program we got down there. Everything had to get sent back and destroyed. And it's a logistical nightmare. And breeding companies now have actually started to screen for known GM events when you provide them with seed. So that seed I've provided to AGT for growing this year, They have their own little chip, which they send off to check that the seed I've sent them doesn't have any of the known components on a vector. But of course, as researchers, we quite often make our own vectors, and so they can be quite different. So at the moment, we are considering anything that is SDN1 as GM, even if we've shown that we don't have any of the bits left in there. And so we stick them out in our GM field site. And this, as far as I'm aware, was the first ever trial of gene-edited barley in Australia. And this is Richard's lines, which I believe you're going to be giving a seminar on a bit later on, and some of the results from that. But so we had this material out in our field site this year. So at the moment, to be safe, we consider anything gene-edited as GM until we can prove otherwise. And I think that's my time. Is that right? So just to sort of finish up, so through the IWIT program, we identified a couple of candidate genes for enhancing yield that, when we put them into GM wheat, did actually have yield improvement in the field. We've now tried to go a non-GM approach to identifying alleles into Australian wheat cultivars. The indications are promising, but we'll find out from this year, from the breeders actually trial this material across Australia. GM field trials are an important part for doing your characterization of your material, but they are very expensive to run and maintain, and the consequences of getting it wrong are huge. And gene editing is a nice way to introduce modifications quickly into plants, but there's still a lot of grey areas and hurdles to overcome before we can even get any of this released. And that just remains for me to thank a cast of thousands involved in the IWIT project, and also the APPN and the Plant SynBio for all the work they've been helping out with the GM field trial. I'll hand over to you, Richard.
Cool. I think we're pretty much on the hour. Thanks again, Stu, for the great seminar, and wrap it up here.